Okay, so we are back with a screencast here with another type of genetic cross. All right, so we are still talking about monohybrid crosses or a one-test cross. A one-test cross only talks about one specific trait. We talked yesterday about tall versus short pea plants. I want to point out to you this is very well handled by your other main source, and that is now chapter 23 in your textbook. All right, so chapter 23, point 1 goes into all of the definitions that we spoke of yesterday: phenotype, genotype, recessive dominant, allele gene, all right, plus the one test cross, which they call one trait cross, and the test cross, okay, and the test cross. All right, so today we want to take a look at what happens when we still have a one trait cross, but it really isn't simple inheritance. This is simple. In a monohybrid one trait cross that shows complete dominance, it's either tall or short. So it's completely dominant. There's no middle here. All right, so dominant trait determines the appearance. Recessive trait has no effect. So if you have a dominant gene, you show tall. That's what we mean by it. All right, and there you can see all of the traits that Mendel worked on in his lifetime. Take a look at the YouTube video on Mendel the other day. Also take a look at the patterns of inheritance there. Pardon the vacuum in the background. All right. So again, one more time before we go into something a little different. Purple, white, purple dominant, white. And then you have purple. All right. So now what happens if something isn't completely dominant? Then you get in science what we call codominance. All right. Complete dominance, tall, short, purple, white. Codominance, it isn't that easy, because in a codominant, what happens is when the heterozygote, the capital letter and lowercase letter, and we don't even use that here because there is no dominant. So when you have the heterozygote, two different alleles of a gene, they either express something completely new or they combine. So let's take a look. Codominance, there are two types, and they're denoted here. All right. Type 1 codominance is when the heterozygote is intermediate between the two homozygotes. So if you look here, you have a red and you have a white flower. It's called a snapdragon. And the red and the white give you pink. It doesn't give you red and white. It gives you pink. And this pink here is considered to be a new phenotype. Okay? A new phenotype. And the new phenotype, let's get an arrow there. So it's not red and white, combination of both, to forget about the color scheme. It's a new phenotype. So the new phenotype, and it's always the heterozygote here. We'll show you how to show that. So in this case, red and white equal pink. So you have a red flower, you have a white flower, and now you can have a pink flower from this. Now this type of, in some quarters, is also called incomplete dominance. They want to keep a track of this term too, because high school uses this term and only calls codominance the next one. But I'm actually using IB lingo, which doesn't use incomplete dominance at all. So it uses codominance and has two types. So keep this in mind. For type 1 codominance can also be called incomplete dominance. So for example, in you, and actually mostly Caucasian people, you have a straight-haired person, which is a trait, and a curly-haired person, which is a trait. And usually that produces children with wavy hair in the heterozygote. So notice 50% wavy in that case, 25% straight, 25% curly. So there's Ollie right here. Sorry, moving on. Sorry, Ollie. All right. Codominance type 2 occurs when the alleles are equally expressed in the heterozygote. No new phenotype. So in this case, you have a red, and it really is kind of a very light kind of brown color in a cow. A red and a white cow give you a red and white cow coat. So notice a red and white speckled cow. All right, notice the different genes there. The head of the cow, different set of genes. The tail also, if we can see a different set of genes. Even the legs, different set of genes. The coat of the cow. So coat type 2 is when they are both expressed equally expressed. Here creates an entirely new phenotype. Here, both expressed completely, but it's still one trait: color of coat, color of flower petal in a plant. So it's a variation. This came along in the later part of the 19th, early 20th century when we learned more about it. Right. So we take a look there. Notice, take a look at this. You have a black chicken and you have a white chicken. Which type of codominance is this? Where you have then the black and white speckled chicken. That would be codominance type 2. Codominance type 2. All right. So in this case here, we want to show them with the screencast how to show this. So this is a little different here because each one of the flower here gets assigned a capital letter. There are no lowercase letters to be used here. A capital letter. So this is how it's shown a little bit different. So if we get a new picture up, right? And we draw you a quick Punnett square. See, look at that. We get you a very quick Punnett square here. Should have had this prepared for you, sorry. Right? And we cross a black cow with a black chicken with a white chicken. All right, a black chicken, since there is no dominant, must be, okay, 2. All right, get that into your notes as must be 2 capital letters. So this is really called homozygous white or black, excuse me, in this case. And then we'll get the white out over here. All right, get another one. All right, homozygous white. So the characterization of codominance, no lowercase letters. Used. No lowercase letters used. So if you cross a black chicken with a white chicken, so the white chicken must be homozygous, but not dominant or recessive. So you're going to get your black and white. All right, clone that 3 other times. You're going to get your black and white speckled chicken. So one of the ways that we can, all right, your black and white speckled chicken. All right, now sometimes you'll also see here, a letter used to denote, like notice black and white, but you'll also see a letter here in the alleles to note what trait it is. So if this was the chicken, you might see an F used. Let's get that capital F, excuse me. An F used. Right? And you see the F always drawn larger, and which means that the lowercase letter is sort of like a superscript here, right? And you see that used very often. So it's a good thing for you to get used to that as well, so that you remember. So it's all about noticing. It's all about noticing which type that you're wanted to be used there. Bring one down. So the correct way to show a cross in codominance, either way. Now, same way with type 1. So if you're looking there, this could very well be black and white flowers give you gray flowers. Only kidding. But if it was, the heterozygote is either the new phenotype or the combination of both. The new phenotype or the combination of both. All right, so you have an example, a worked example of how to show it. How would you show the genotype and phenotype here? Well, there are 3 possible genotypes, right? Homozygous, black, homozygous white, which I'm going up to. And heterozygous. Now you can and heterozygous. Now you could also use letters capital B, capital W, and BW capital, all capital. And the genotype, or the phenotype is the same. You would say black, white, and if it's a new phenotype, gray, and if it's a combination, black and white. All right. So it is a variation of the main cross. All right. So I want to listen to the YouTube video. Sorry if I babbled on a little bit, but trying to conduct the class like we have in the class. All right. So done with codominance.